Molecular genetics is a scientific discipline concerned with the structure and function of genes at the molecular level. It uses the tools of molecular biology, including genetic engineering, which is the direct manipulation of an organism's genome. The essential aim of molecular genetics is to relate how changes in the structure and sequence of genes results in functional changes in protein expression, function, and ultimately health and disease. Hereditary information is encoded in the chemical language of DNA and reproduced in all the cells of our body. It's the DNA program that directs the development of our biochemical, anatomical, physiological, and to some extent, behavioral traits. Molecular genetics is a branch of genetics that investigates the chemical and physical nature of genes and the mechanisms by which genes control development, growth, and physiology. Molecular genetics differs from classical genetics in that it treats genes and their products as chemicals rather than abstract things. Several lines of discovery prior to the 1950s led to the birth of molecular genetics. Biochemical studies of genetic traits, which ultimately supported the one gene, one enzyme hypothesis. The discovery of chemical mutagenesis, which showed that genes could be permanently modified by reactive compounds. The investigation of the chemical makeup of genes, culminating in the demonstration that DNA had the properties expected for genetic material. The discoveries of structural molecular biology, in particular the Watson and Crick proposal for the structure of DNA, provided the foundation of molecular genetics in subsequent decades. So the modern study of genetics at the level of DNA is known as molecular genetics. Molecular genetics has a broad reach. It helps us determine the pattern of descendants, understand how genetic mutations cause disease, how certain traits are passed on to subsequent generations, and how patterns of relatedness occur across these generations. It offers the healthcare community an understanding of how genetic variability of individuals may impact risk factors for disease, but it also plays important roles in infectious disease, as we're witnessing today. As we've said, molecular genetics utilizes the tools of molecular biology. These techniques allow us to manipulate and analyze DNA. Some of those techniques allow us to sequence the DNA. Sequencing DNA tells us the precise sequence of nucleic acids, literally the order of nucleotides that make up a piece of DNA. We can also use a variety of molecular tools to then digest DNA, separate it, change or mutate it, amplify it, and analyze it. But let's start with sequencing. DNA consists of a linear string of nucleotides, or bases for simplicity, referred to by the first letters of their chemical names. A for adenine, T for thymine, C for cytosine, and G for guanine. The order of nucleic acids in polynucleotide chains ultimately contains the information, heredity, and biochemical properties of all terrestrial life. The ability to measure or infer such sequences is imperative to biological research, and in no small part is the starting point for understanding much about our biology. Watson and Crick famously solved the three-dimensional structure of DNA in 1953, working from crystallographic data produced by Rosalind Franklin and Maurice Wilkins. This contributed to a conceptual framework for both DNA replication and encoding proteins. However, the ability to read or sequence DNA did not follow for a long time. The process of deducing the specific order of nucleotides in DNA is called DNA sequencing. Since the DNA sequence confers information that the cell uses to make RNA molecules and proteins, establishing the sequence of DNA is key for understanding how genomes work. The first sequencing efforts were directed at elucidating readily available populations of relatively pure RNA species like microbial ribosomes or transfer RNA, or even the genomes of single-stranded RNA bacteriophages. Progress was made in the mid-1960s, but it was slow going. In the early 1970s, it was observed that Enterobacteriophage lambda possessed a 5' overhanging cohesive end. Ray Wu and Dale Kaiser used DNA polymerase to fill in those ends with radioactive nucleotides, supplying each nucleotide one at a time and measuring the incorporation to deduce the sequence. It wasn't long before the principle was generalized through the use of specific oligonucleotides to prime the DNA polymerase. Incorporation of radioactive nucleotides could then be used to infer the order of nucleotides anywhere, not just at the end termini of bacteriophage genomes. However, the actual determination of bases was still restricted to short stretches of DNA and typically involved a considerable amount of analytical chemistry and fractionation procedures. The major breakthrough that forever altered the progress of DNA sequencing technology came in 1977 with the development of Frederick Sanger's chain termination, or dideoxy technique. The chain termination technique makes use of chemical analogs of the deoxyribonucleotides, also known as DNTPs, where the N is simply standing in for either an A, T, C, or G. These deoxyribonucleotides are the monomers of the DNA strands. Dideoxynucleotides, or DDNTPs, lack the 3' hydroxyl group that's required for extension of DNA chains, and therefore they can't form a bond with the 5' phosphate of the next DNTP. Mixing radiolabeled DDNTPs into a DNA extension reaction at a fraction of the concentration of the standard DNTPs results in DNA strands of each possible length being produced, as the dideoxynucleotides get randomly incorporated as the strand extends, halting further progression. By performing 4 parallel reactions containing each individual DDNTP base and running the results on 4 lanes of a polyacrylamide gel, one is able to use autoradiography to infer what the nucleotide sequence in the original template was, as there will be a radioactive band in the corresponding lane at that position of the gel. One can then simply read the DNA sequence, starting with the shortest DNA strands at the bottom and reading upwards as the equivalent of going from the 5' to the 3' direction. For example, in these cases, as we read upwards, we can see that the DNA sequence here is adenine, guanine, guanine, adenine, and this was replicated twice. The next sequence is going to be a cytosine, guanine, 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 thymine, guanine, cytosine, thymine. And simply by reading the sequence, we're able to deduce the DNA sequence. While working on the same principle as other techniques that are producing all possible incremental length sequences and labeling the ultimate nucleotide, the accuracy and robustness and ease of use led to the dideoxy chain termination method, or simply put, the Sanger sequencing method, to become the most common technology used to sequence DNA for years. The technology for DNA sequencing was made faster and less expensive as part of the Human Genome Project. Automation replaced the manual sequencing method with development of fluorescent labeled ddNTPs. Slab gels were still poured, however, all 4 bases were combined into a single reaction and loaded together. Samples would be electrophoresed and separated on the gel. Once the amplified products reached a region on the bottom of the plates, a laser would excite the fluorescent labels and the color could be recorded by a camera. The resulting images were collected and computer analyzed as shown on the left. While this increased sequencing capacity fourfold, a significant amount of sequencing was still being performed that required pouring slab gels. And these plates and their electrophoresis required at least 12 hours or longer. The development of capillary sequencing occurred in the 1990s. This started with a single machine, and the new technology eliminated any need for pouring gels. Instead, a semi-liquid polymer was injected into the capillary before each run. Individual runs reduced the length of time required for the electrophoresis to less than 3 hours. Capillary sequencing was faster and much more sensitive. It required much less DNA to be added to the PCR amplification, and better chemistries were developed in conjunction with automated sequencing. Capillary sequencers today characteristically can perform anywhere from 4 to 96 samples in a single run, and the run typically requires 2 to 2.5 hours to complete. Now that we understand the importance of knowing the sequence of nucleic acids in a DNA strand, we can also look at the ways in which we can manipulate those DNA strands. Restriction enzymes are enzymes that cut DNA at specific sequences called recognition sites. Restriction enzymes were first identified in bacterial cells like E. coli. The bacteria use these specialized enzymes to defend themselves against viruses that would inject their DNA into the bacterial cell. The restriction enzymes were therefore a kind of biological defense mechanism that would cut up foreign DNA into fragments to prevent viral infection. The names of restriction enzymes are derived from the genus, species, and strain designations of the bacteria that produce them. For example, EcoR1 is isolated from E. coli, strain RY13. Eco refers to the genus and species, the first letter of the genus and the first 2 letters of the specific epithet. R is the strain of the E. coli, and the Roman numeral 1 indicates that it's the first enzyme of that type isolated from E. coli RY13. You can think of restriction enzymes as a kind of molecular scissor that cuts DNA at specific sequences of DNA base pairs. Each type of restriction enzyme seeks out a single DNA sequence and precisely cuts it at one place or a set distance from that site. For instance, the enzyme shown here that we're going to use as an example throughout this presentation, EcoR1, cuts at the sequence GAA-TTC. It cuts between the G and the A. A second enzyme, DNA ligase, may then be used to reassemble the pieces at any desired order. Together, these 2 types of enzymes allow researchers to assemble customized genomes. Over 400 restriction enzymes have been isolated from bacteria, and each has its own unique recognition site that it cuts at or near. One question you may have is how are bacteria immune to their own restriction enzymes? It turns out that bacterial restriction sites are highly methylated, making them unrecognizable to the restriction enzyme. Now, different restriction enzymes have different stringencies. So a highly stringent restriction enzyme only cuts at that specific sequence. By changing the conditions that the enzyme is exposed to, you can make restriction enzymes less stringent so that they may be less specific in the way that they cut the DNA. An interesting property of restriction enzymes simplifies this molecular cutting and pasting. Restriction enzymes typically recognize a symmetrical sequence of DNA, such as the site of HinD3, which is shown on this figure. Note that the top strand is the same as the bottom strand read backwards. When the enzyme cuts the strand between the A and A, it leaves overhanging ends. These are termed sticky ends because the base pairs formed between the two overhanging portions will glue the two pieces together, even though the backbone is cut. Sticky ends are an essential part of genetic engineering, allowing researchers to cut out little pieces of DNA and insert them into specific places where the sticky ends match. Not all restriction enzymes leave sticky ends, however. Some restriction enzymes, like EcoR5, also cut DNA, but they form blunt ends without single-stranded tails. These can also be inserted into target DNA via the action of DNA ligase. But DNA ligase isn't picky. It can't tell the difference between foreign and host DNA. And this enables the creation of chimeric DNA, which is DNA from two separate sources. When a restriction enzyme cleaves a restriction site, it's going to create those sticky ends we've just described. And this is really useful. So vectors are basically circular pieces of DNA that biotechnologists can use and modify. By cutting open vector DNA with the same restriction enzymes used to cleave the target DNA, we can create complementary sticky ends. This facilitates the insertion of the target DNA into the vector. The fragment is then glued in place with DNA ligase, which creates a phosphodiester bond necessary to complete the sugar phosphate backbone of the now newly transgenic DNA. Now, there can be numerous recognition sites for a given enzyme on a strand of DNA. The restriction enzyme is not going to select one restriction site as opposed to another. It's going to digest the DNA at each one of those sites. So if we add the EcoR1 restriction enzyme, and remember, it has the recognition sequence of GAATTC to this specific example, how many recognition sites are there? There are one. That's shown here, and it would cleave between the G and the A. And there's a second site. So how many fragments would we get? If we have two restriction sites and it's a linear piece of DNA, we would expect to get 3 fragments. Now, in this case, the length of each fragment is going to depend on the distance between the restriction site from each other and the distance of the restriction sites and the ends of the DNA strand. So we would get 1 7 base pair fragment, a 23 base pair fragment, and a 15 base pair fragment. If we use restriction enzymes to digest genomic DNA, they're going to make numerous cuts and generate little fragments of DNA of all different sizes. We can then visualize those fragments by running our digested DNA sample on an agarose gel. And each of those bands is going to be a DNA fragment of a specific size. So My question to you is, we're loading our DNA into our sample gel, into the wells of our gel, and we're going to apply an electrical charge to the buffer. So is DNA positively or negatively charged? DNA is negatively charged, and therefore it's going to travel through the gel towards the positive pole. Restriction fragment length polymorphism analysis, or RFLP, is a molecular method of genetic analysis that allows individuals to be identified based on unique patterns of restriction enzyme cutting in specific regions of DNA. RFLP analysis involves cutting a particular region of DNA with a known variability with restriction enzymes, and then separating the DNA fragments by gel or electrophoresis, and determining the number of fragments and their relative sizes. Every person has an entirely unique fragment pattern that's much like a molecular fingerprint. Some of the applications of RFLP include DNA fingerprinting. Forensic scientists may use RFLP analysis to identify suspects based on evidence samples collected at scenes of crime. We can also use it to determine paternity for tracing ancestry. It can also be used in studying evolution and migration of wildlife by studying breeding patterns in animal populations, as well as the detection and diagnosis of certain diseases. So here we're looking at two sample DNA sequences from two different individuals. The yellow highlighted regions are going to be the recognition sites for EcoR1, and we see that person 1 has 2 recognition sites for EcoR1. Person 2 has only 1 of those recognition sites. The second site has an alteration genetic sequence that negates the recognition sequence for EcoR1. So when digested with EcoR1, the sample from person 1 should result in 3 different size DNA fragments, but the sample from person 2 will result in 2 different size DNA fragments. So what do we do with this? The restriction digest must be run on an agarose gel in order to determine the number and size of the DNA fragments that are produced by the restriction digest. The DNA sample is going to be mixed with a loading dye that, among other things, contains bromophenol blue and glycerol. The glycerol is going to help to weigh the DNA down as it's loaded into the wells to prevent it from diffusing into the buffer solution. The dye helps us keep track of how far the DNA fragments have migrated in the gel in order to prevent the DNA from running out of the bottom of the gel. We're then going to turn on the electrical current, and the DNA is going to migrate through the electric field, effectively separating the fragments by size, with the smaller fragments migrating through the gel more quickly. The gel is then stained in order to visualize the DNA fragment. So we can use this technology for a forensic technique called restriction fragment length polymorphism analysis, or RFLPS, colloquially called DNA fingerprinting. So remember that the restriction fragment length polymorphism is a type of polymorphism that results from a variation in the DNA sequence recognized by restriction enzymes. And these are markers on genetic maps. So if 2 individuals, like we see here, have different RFLPS, then there's going to be a difference in the number of DNA fragments that we can observe between these individuals. So to go back to this example, what would we expect our final gel to look like in this RFLP analysis? Well, if we start out here, the first thing we're going to want to have is a molecular marker. The molecular marker, or ladder, is made up of DNA fragments of known length. And we use this to compare to the samples so that we have a good idea of the size of the samples that have been loaded. We then want to run the fragments or the DNA sample from person 1. We expect 3 fragments, a 7 base pair fragment, a 15 base pair fragment, and a 23 base pair fragment. For person 2, we expect a 15 base pair fragment and a 30 base pair fragment. What's clear is that the patterns produced by this analysis are very different and are easily discernible by using this molecular technique to distinguish these two individuals. Some applications for RIFLIP analysis include the DNA fingerprinting that we just described, paternity, and we've also talked about genetic diversity. The technique of using RIFLIP detection for variation in genomes is actually a vital tool in genome mapping and genetic disease analysis. If the location of a particular disease gene is being sought in a certain chromosome, then researchers can analyze the DNA of members of a family with the disease, looking for similar patterns of inheritance in RIFLIP alleles. Once a disease gene is localized, conducting RIFLIP analysis on other family members could reveal a carrier of the mutant gene or signal overall disease risk. It's important to note that the RIFLIP technique is not widely used now that newer, more robust techniques are used for DNA analysis in both forensic science and genetics. So, for example, much of RIFLIP analysis has now been replaced by PCR. So, some cons. Basically, the RIFLIP analysis technique is tedious and slow. Aside from that, it also requires a large amount of the starting sample DNA, and that basically has to then be isolated, which can also be laborious.